I'm going to talk a little bit about some work we've been doing trying to understand genotype-phenotype relationships in bladder cancer, and our relationship with Mission Bio and the use of the Tapestri has really been important in pushing this work forward. So just as a brief overview, this is a slide showing the putative origins and pathogenesis of bladder cancer. Bladder cancer can be divided into two major stages. One is a non-muscle invasive stage, and the other is a muscle invasive or advanced stage of disease. And so this is just a depiction of the epithelial cell layers within the bladder. And so like many other epithelial tissues, there is a basement membrane, basal cells that essentially line the basement membrane. And then as you get towards the bladder lumen, you have intermediate cells and then some of these luminal-like umbrella cells. And so it's thought that there are both basal and luminal bladder cancers. And so one of the ideas is that basal cancers, or those that take on more basal squamous differentiation, arise from these basal cells, whereas some of the luminal type cancers arise from the luminal cells that are closer to the bladder lumen. And this is important because it's thought that basal cells give rise to the muscle-invasive types of bladder cancer, and the luminal cells give rise to more of the superficial, non-muscle-invasive bladder cancers. However, this doesn't mean that these luminal cancers can't become muscle-invasive. In fact, these low-grade luminal tumors can take on additional genetic alterations and potentially convert into more invasive carcinomas as well. And there's been increased appreciation that there is, in fact, plasticity between these cancer states. And so this depiction may be a little bit simplified. One of the key features of bladder cancer is that there is a very high mutational burden. And so in TCGA studies looking at muscle-invasive bladder cancer, one can appreciate that in this plot, we're looking at individual tumors on the depicted in each of the columns, and then each of the rows represents a gene that's altered within individual tumors. And so you can see that if you just look down each of these columns, there's quite a bit of heterogeneity. And if one were to tabulate the number of genetic alterations that occurs in each of these tumors, you'd find that over 30 genetic alterations occur at a frequency of greater than 5%. And on average, each tumor has maybe about 5 genetic alterations within this set of 30 genes. And so that's the genotypic complexity, and then we think about the phenotypic complexity. And so there's been a push towards a molecular classification of bladder cancers, and a variety of groups have actually engaged in this endeavor. And there was recently a consensus molecular classification that was published. And so you can see that there's a basal squamous group, a neuroendocrine-like group, a stroma-rich group, a luminal papillary group, luminal unstable, and luminal non-specified. And then on top of this complexity, there are a variety of histologic variants. So most bladder cancers are urothelial cancers, but there can also be histologic variants that can coexist within these tumors. So for instance, there can be sarcomatoid carcinomas, small cell carcinomas, plasmacytoid carcinomas, and micropapillary variant carcinomas. And it's not entirely clear how the genetics necessarily plays into the emergence of these variants. And so historically, the technologies that have been used to address this complexity in a genetically defined manner have included many pre-genomic era technologies. So we've certainly relied upon genetically engineered mice for decades, the use of human cancer cell lines and patient-derived xenograft models, and then the use of bulk sequencing to characterize the genetics of these model systems. There are some issues related to these. So we've used genetically engineered mice now for about 3 decades, and there are relatively few models of bladder cancer that really recapitulate the diversity of bladder cancers that we see in humans. Certainly these are slow and very resource intensive. The use of these human cancer lines at PDXs, many of them are not genetically defined, and they can evolve over time in the laboratory. And so to address this, we wanted to try and develop a system where we could use a very scalable, economical approach to try and generate much of this diversity and recapitulate it in the laboratory. And so we were going to rely heavily on organotypic or organoid cultures, which are scalable and enable facile ex vivo manipulation. And then to incorporate single cell sequencing so that we can do massively parallel analysis. And so I'll tell you a little bit about the strategy that we've taken. As a general approach and overview, this is sort of the idea that we took on and moved forward. And so we wanted to model higher order genetic interactions in tumor initiation by using high lentiviral multiplicity of infection of multiplex perturbagen pools. And so if we have a lentiviral library of genetic perturbagens, those that mimic gain of function and loss of function, and we have target cells, the idea is to introduce these genetic perturbations at high MOI, and so that essentially what will happen is you'd randomly distribute combinations of these perturbagens into individual cells. Here we describe these as primary epithelial cells. Once phenotypic selection has occurred, so in this case we are looking for the perturbations or combinations of perturbations that induce tumorigenesis, then we can take the cancer cells and perform targeted single cell DNA sequencing or single cell RNA sequencing to really then associate compound genetic perturbations with these complex phenotypes. And you can appreciate that although our initial focus has been on tumorigenesis, one can easily replace the target cell population, the type of phenotypic selection, and the ultimate cells that are being analyzed. For instance, we also have a project in the lab where we're looking at cell reprogramming. So you can think about cell state 1, selection for cell state 2, and then trying to figure out what are the genetic perturbations that induce that second cell state. So getting back to bladder cancer, this is the general functional genomics approach that we've taken. And so we have generated a library of lentiviral vectors that recapitulate 32 gain of function or loss of function events. And so these lentiviral vectors have encoded within them unique 5' and 3' barcodes. And so the difference between these two constructs is that the first construct is meant to overexpress an open reading frame, whereas the second construct overexpresses a short hairpin RNA. And so over on the right, you can see the various gain of function and loss of function genes that we are studying. And these were selected just based on the frequency at which they occurred in human bladder cancer. And we set a general cutoff of around 5%. And so the idea is to take basal bladder urothelial cells from mice, use this lentiviral pool to transduce these basal bladder urothelial cells to, for a short period of time, culture these in organotypic cultures, to then take these out after a couple of days, recombine with a stromal element. This is embryonic bladder mesenchyme, and then we subcutaneously graft these in mice. And the idea is that in mice, we'll biologically select for tumorigenic clones, the hope being that these clones will be, or these tumors will be polyclonal. And then we can use single cell DNA barcode amplicon sequencing to really determine what are the lentiviruses that have been integrated into each of these tumor clones. And this is just an example that is shown here. And so we can also go back and then compare the population that we started out with, so we can do bulk DNA barcode amplicon sequencing just to show that we have good representation, and then after tumorigenesis we can determine what are the genetic alterations that are enriched in the tumor. So obviously this was a pretty lofty goal, and so we had to do quite a bit of feasibility analysis just before we could move on. So one of the major things that we had to determine was whether we could actually achieve high multiplicity of infection in mouse urothelial cells in organoid culture. And so we dissociate mouse bladders from C57 black 6 mice, and then we can transduce these. And so depicted here are untransduced urothelial organoids. And so we transduce with the lentivirus expressing GFP. So there's no GFP signal visible here. Whereas when we add 5 microliters of concentrated GFP lentivirus to each organoid well, you can see that the majority of these are now expressing GFP. And so if we titrate the amount of virus that we add and then measure the GFP frequency by flow cytometry, we can see that this is relatively linear. And with exceedingly small amounts of lentivirus, we can achieve pretty much a complete transduction. But more importantly, when we look at the GFP copy number, we can achieve up to 20 copies per cell, which certainly is high MOI transduction. The next thing that we wanted to determine was whether we could really enumerate cell populations marked by these compound lentiviral barcodes using a custom panel that we generated with Mission Bio on their Tapestri. And so this is a depiction of a lentiviral construct. And so the custom panel essentially amplifies the barcode portions of this GFP, the 3' barcode, and then we also included several endogenous mouse genes as well into our custom panel. And so to start, we use 3T3 cells. And so this is a mixed 3T3 cell population. About 29% of them were unlabeled. 14% had 4 barcodes. 1.5% had 3 barcodes. Close to 60% had 2 barcodes, and then another 2 barcodes at a frequency of 0.14%. And so when we analyze this on the Tapestri, we analyze about 2,000 cells, and what we can see is that we get pretty close to the expected frequency through the use of the Tapestri. And so this really gave us a lot of confidence to move forward. The last thing that we needed to do was really to optimize the generation of our lentiviruses. And so one could anticipate that if you have barcoded open reading frame lentiviruses and short hairpin RNA lentiviruses, that the size of these constructs will actually affect the production of lentiviral virions. And so what we did was we initially generated these lentiviruses individually in arrayed format. And we did this to prevent barcode swapping, which is a fairly well-known phenomenon in this field. We then pooled and concentrated the lentiviruses by ultracentrifugation, transduced the primary bladder urothelial cells in organoid culture, isolated genomic DNA, and then amplified our lentiviral barcodes and generated libraries by two-step PCR. And so when we did this and performed sequencing, as expected, the initial average of open reading frames to shRNAs was something like 0.1, really indicating that shRNA lentiviruses are made much more efficiently than these open reading frame lentiviruses. And so we use this information to then adjust the producer cell surface areas, went through this process again. And by doing so, then we could nearly normalize the production of these lentiviruses such that the ratio of open reading frames shRNA was now 0.8. So having established these various parameters, we decided to go forward. And so I'm going to share with you today just one vignette of the type of data that we're able to generate with this system. And so using the pool introduced into these mouse urothelial organoids and then transplanted into mice, we have generated several tumors. This is one tumor in which we can actually identify 3 concurrent cancer histologies, and these are clinically relevant cancer histologies. So in this region of the tumor, you can actually see papillary carcinoma with inverted growth. In this region of the tumor, you can see urothelial carcinoma with squamous differentiation. And then here, you can see sarcomatoid carcinoma. And so the immediate question here is, do these represent 3 clonal cancer outgrowths, or are these really divergent, differentiation of a common precursor? And so we turn to the Tapestri to try and guide us towards an answer. And so we took this tumor dissociated to single cells and performed single cell DNA amplicon sequencing, and we were able to resolve 3 dominant clones within the tumor. And so clone 1 represented about 39%, clone 2, 21%, clone 3, 7%. And so about 3,300 cells were analyzed in this experiment. The important thing to see here is that there are a set of open reading frames that have been introduced commonly to all 3 of these clonal populations. And then presumably, these guys out here are going to be specific to each of the individual dominant clones. And so we were really struck by FGFR3. It's very well known that FGFR3 mutations and activated signaling are enriched in luminal papillary urothelial carcinomas. And so we wondered whether this was really driving that papillary carcinoma histology that we saw previously. And so we wanted to take these common alterations and then add FGFR3 to see whether they could recapitulate that phenotype. And so this is the experiment that we did. Again, we went through the process, but this time, instead of introducing that larger lentiviral pool, again, we added mutant FGFR3 and those 4 common genetic alterations. And we wanted to see whether, if we go through this entire process, do we generate papillary carcinoma histologies? And so the answer is yes. So this is what the tumor looks like. It has this papillary architecture. It's got a consistent staining pattern here. And so really the question is to nail home whether in fact this is driven by FGFR3. We're doing some leave one factor out transformations where we will leave out FGFR3 but keep the other 4 and then the various combinations thereafter. And those are currently in mice now. So another question that we asked is this completely cell intrinsic? And so because our model system requires the addition of EBLM, embryonic bladder mesenchyme, we asked, if we remove that, will we also obtain a papillary carcinoma histology? And so we repeated this. And so what we find is that in the absence of EBLM, the tumors that we actually get out are actually squamous carcinomas. And so you can see that pretty clearly here with sort of this onion peel type squamous differentiation. And so this really leads us to question whether the lack of cues for basal to luminal differentiation within this model system, which are presumably derived from EBLM, really leads to squamous cancer differentiation. So having established these models of both papillary carcinoma and squamous cell carcinoma, using the same genetic alterations, but obviously derived within different environmental contexts, we were able to take these, put them into organoid cultures, and we asked, okay, there is a FDA-approved FGFR inhibitor, erdafitinib, for urothelial carcinoma. Is there a difference in sensitivity to erdafitinib with these 2 model systems? And in fact, we see that at the 10 micromolar dose, that the papillary carcinomas actually are much, much more sensitive to erdafitinib than the squamous variety of this carcinoma. FGFR3 is in fact expressed in both models. So, really one of the questions that we're asking now is, how does cancer lineage potentially affect FGFR3 signaling, if at all? And so we're actually doing a lot of analyses downstream of FGFR3 within these model systems to determine whether there are differences in signal transduction and effects downstream. And so this is just one vignette that I wanted to share with you today. But certainly we have other tumor models that we're also evaluating, in which the Tapestri has been very helpful in actually resolving not only the clonal architecture, but also really providing a link between the genotype and the phenotype within our functional studies. So I just wanted to acknowledge a very talented technician in my lab who's done pretty much all of this work, Alicia Wong. And then the bladder cancer group here at Fred Hutch and the University of Washington. Certainly, Mission Bio was very, very helpful. They worked with us to generate this custom panel very early on. We're very excited to apply this sort of technology to mouse models of cancer. And these are my lab funding sources. So with that, I'll stop and take any questions.
Great. Thank you so much, Dr. Lee. We really appreciate you taking the time and explaining your research. We have a couple really great questions that have come through. So you made a lot of different mutations in mouse models. Curious if it recapitulated what you saw in humans in terms of the mutations, and were there any surprising results?
Yeah, I mean, we're still early in on this project. So what we want to do eventually is to collate all of the data that we get from all the tumors that emerge, and then really try to see whether we can, within our tumors, whether that sort of mimics the frequencies and associations that we see in human tumors. Unfortunately, we don't have a large enough N to do that just yet, but ultimately that's a goal.
Great. This is a personal question. So we've released DNA plus protein on the platform. Do you see, you talked about cell states. Do you see the protein capability adding to this research, and how would you use it?
Yeah, absolutely. I think, bladder cancer, because of the variety of histologic variants and the importance of cancer cell differentiation states, of which there are many, we know obviously that cell surface markers and other protein expression really indicate the cell differentiation states in cancer. And so that might be helpful to us in terms of understanding, the association between the genetic alterations that we introduce and how that necessarily affects cancer differentiation downstream. And so the protein can certainly help with that level of analysis, as can single cell RNA sequencing capabilities.
Great. Question, how great is the mortality in these mice?
So we are using subcutaneous models. And so we have generally been euthanizing the mice once they achieve sort of the Iacocca maximal tumor volumes that are established here. We recognize that, there are a couple of issues with the model as is, but this is just a start. Ultimately, what we'd like to do is to do orthotopic grafts in immune competent models. And so we're working towards that. We've already established that, we can take these cells that are transduced with these lentiviruses that express GFP, and we can transplant them into syngeneic immune competent mice. And so the next step really is to acquire the technical skills to actually do the orthotopic injections, which is not trivial. So we have some collaboration set up to get that going.
Got it. So a technical question. You showed your organoid and that the 3 different regions had different cell types. Did you take different sections of your organoids and do single cell, or was it one section and you resolved the 3 different populations using the single cell sequence?
So essentially, yeah, we cut the tumor in half, and then we essentially took half of it for histology. And then the other half we dissociated to send off or to perform the Tapestri analysis. What we're doing now, because we don't get in situ level clonal analysis, obviously, when we're sort of dissociating single cells, is we're actually doing laser capture microdissection, and then we're amplifying the barcodes from each of the regions and sequencing those to confirm that, in fact, the FGFR3 mutant corresponds to the region that looks like papillary carcinoma. But obviously, our validation experiments suggest that that is, in fact, the case, but it's just good to have that as a confirmation as well.
Great. It'll be great to add LCM experiments to those. Question here on the organoid. Were the differences in squamous and papillary phenotype of cancer recapitulated in the organoids?
Yeah, so we have preliminary data looking at Western blot analysis. And so we do have various markers of sort of luminal differentiation and squamous differentiation. And in fact, they are maintained within organoids. We have not passaged them, multiple times in organoids because there is some recent literature indicating that there can be quite a bit of plasticity where you can switch from one cell state to the next. But that's something that we'll examine as we continue to passage the organoids.
Great. Another technical question. The clonal analysis using Tapestri identified 3 clones, 39%, 21%, and 7% frequency. What about the rest of the 33% of the cells? Yeah,
so they were basically a number of very, very rare clones that were generally under 2%. And so, we do see that in some of our analyses. I think that just relates to the fact that we are able to create a wide variety of combinations within the tumors. And so, it's hard to know what to make of that just yet, but that is an issue that we're trying to address. For now, we're sort of looking at the low-hanging fruit, right? Sort of the more common alterations that emerge from our analyses. But ultimately, I think it would be helpful once we can collate all of these data together to look to see whether there are any other insights or associations that we can derive from the entire data.
Great. And then just one final question. I know we're in really unprecedented times with COVID. We were just wondering how you're doing. How's your lab doing? Are you up and running?
Yeah, so the Fred Hutch was pretty proactive about ensuring safety, and so we went. Our labs pretty much shut down. There were only essential personnel through much of March, April, and May. And then in May, we started to dial up a little bit very slowly. And we've had programs here to really screen for symptoms, and then also there's a study here called Back to Work, in which employees can enroll in a study to assess for antibodies to COVID. And so I think the administration here has been very careful, and just last week, we're actually up to full speed again in our labs. Obviously, it's not the same as it was prior to all this in that we're all wearing masks. We're trying to reduce density as much as possible, so people are working these staggered schedules. But at least from what I've heard from administration, there hasn't been any upturn or spike in new cases of COVID within employees here. So that's certainly very promising.
That's great to hear. I know Seattle was sort of the start for the US. So it's really great to hear that you're up and running. One more final question that's come in. Did you look at copy number variation in these studies?
So we did not really look at copy number variation. I think that's an interesting point. Our panel is specifically looking at the barcodes and then these endogenous mouse genes. We're not really anticipating, just based on prior studies, that there will be significant copy number variation within the tumors that we generate. Certainly there can be copy number variation in terms of the number of lentiviral copies that are introduced per cell. But there are things that we can't control here. For instance, we're expressing these shRNAs from a U6 promoter. We're expressing ORFs from a ubiquitin promoter. It's not going to genocopy the addition of one more copy of a gene, right? I mean, you're really blasting the expression of these ORFs. And so, we didn't think of that would necessarily be a priority just because some of this is artificial, but that's something that could be considered in the future.